Hello, and welcome to Virtual Investor Conferences. My name is Anthony Kraus, and on behalf of OTC Markets, as well as our co-host, Zach Smallcap Research, we're very pleased you have joined us for our next live presentation from Aethlon Medical. Their session will be moderated by Marla Marin, Senior Analyst at Zach Smallcap Research. Please note, you can submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings through the Schedule Meetings tab found on the conference platform. At this point, I am very pleased to welcome Jim Frakes, Interim CEO and CFO, and Stephen LaRosa, MD, Chief Medical Officer of Aethlon Medical, Inc., which trades on Nasdaq under the symbol AEMD. Welcome, Jim, Stephen, and Marla.
Thank you. Hi, Jim. Hi, Steve. Thanks for making time for this. Let's start out by getting a little bit of information about the basics. What is the Hemo Purifier?
Hi, Marla. So the Hemo Purifier is displayed. I think you can see it in front of you. It's an extracorporeal medical device. It's an investigational device meant to be inserted within a veno-venous circulation via a blood pump. And it really composes three aspects. It separates plasma, size excludes molecules less than 200 to 500 nanometers, and it affinity binds. And so the way it's displayed in front of you, you can see it has two ports at the end. That's where the blood enters and exits. The blood enters the device and goes into the lumen inside those fibers, which is the white material that you can see. And then it has pores of approximately 200 to 500 nanometers. Molecules and plasma circulate through those pores to the area around it. And if you can see in the picture, there is the outside those fibers is our proprietary affinity resin, which is composed of a medical grade diatomaceous earth that's covalently bound to a plant lectin known as GNA. And that's really the special sauce of the device. As the plasma traverses the outside of those fibers and encounters the resin, structures that have a sugar called mannose on their surfaces, which is seen in envelope viruses and extracellular vesicles, are bound by the GNA and removed from the plasma. The plasma, as it comes across the device, then will re-enter the lumen of the device to rejoin the cellular elements, the red blood cells, white cells, platelets, and then go back to the patient so that nothing is lost, no plasma has to be administered to the patient. It has breakthrough device designation in both life-threatening viral infections as well as in oncology.
Thank you. And I know you've done a lot of work in terms of preclinical research on the efficacy of the Hemo Purifier. Can you talk a little bit about that, what your preclinical database looks like?
Sure. So we have both a preclinical and a clinical database. It'd be important to mention on the clinical side of things that we do have a safety database that involves 38 patients that have been treated in 164 unique hemopurifier sessions. And the device has widely been tolerated quite well. To answer your question about the preclinical side of things, there's kind of two aspects to preclinical testing. One is safety, and we've gone through all the standard preclinical testing, which includes genotoxicity, biocompatibility, hemocompatibility. So we have that database. The other part of preclinical data is really non-clinical performance of the device. And I'll speak briefly about three areas, focusing lastly on oncology, which is the company's main focus. So the first is in virology. So as I said, we're in life-threatening viral infections. Most enveloped viruses are coated in a sugar substance, including mannose, which is the target for our device. So we've tested a wide array of enveloped viruses against our device, utilizing a miniature version of the Hemo Purifier that you see there. And we have seen a broad spectrum removal of envelope viruses, including some timely important viruses that people are concerned about, such as dengue and other flaviviruses. Bird flu, which has been in the news a lot lately, H5N1, has been removed in vitro. Dengue, which we're seeing cases now domestically acquired in the US, also removed, as well as SARS-CoV-2, both the spike protein and the many variants have been removed. So we have a broad panel of envelope viruses. I think that's important because it's likely that if there's going to be another outbreak or pandemic, it's probably going to be with an enveloped virus, and we would stand poised to test this new virus in vitro, and then go to the next step of clinical trials if the preclinical data bore that out. So I think that's important. I'll briefly mention our preclinical work in transplant. Organs, when they're recovered from from donors prior to going into recipients, are typically put on a machine perfusion circuit to maintain their viability. What's been noted is that extracellular vesicles are released from these organs and play a role in delayed graft function in the recipient. So with that in mind, we actually did some in vitro work with perfusates following machine perfusion of organs and ran them over our Hemo Purifier in our lab and showed reduction in both extracellular vesicles and their cargo microRNAs that are implicated in poor function. And we just posted that on the preprint server BioRxiv. So that's virology and transplant. But our focus is on oncology. So I want to talk about the preclinical data in oncology. There are basically three main sources of data that have supported the clinical trials, which I'm sure we'll talk about. One is we have an experiment where we got preclinical samples from patients on a number of different tumor types, including breast, esophageal, head and neck cancer. And we ran them over, we took those samples, we isolated the extracellular vesicles out of the samples, put them in a buffered solution, and ran them over a miniature version of our Hemo Purifier. And regardless of the tumor type, we saw between 92 and 99% removal of the extracellular vesicles during that circuit. And that data was presented in 2020 at the AACR meeting. We followed that up more recently. We thought it was important to demonstrate that we're removing extracellular vesicles directly from plasma, since that's the most relevant to a clinical trial. And so we actually got plasma from a patient with non-small cell lung cancer, ran it over a miniature version, and actually confirmed that we can directly remove the EVs from plasma itself, not EVs in plasma. The third piece of information that helps support our trial is we actually did, in compassionate use, emergency use, we did 8 treatments on a patient with severe COVID. Now, this patient didn't have malignancy, but what was important in this is that we were actually able to measure extracellular vesicle levels in that patient over the course of the 8 treatments, and showed reliably that over the course of the 8 treatments, the extracellular vesicle concentration went down, as well as microRNAs that were involved in organ function. So that was our first demonstration that we could actually remove extracellular vesicles in the in vivo setting. So with those 3 pieces of information, we were satisfied that we had good preclinical data to support going into the clinic.
Steve, we probably should define EVs and exosomes a bit.
Great point. So extracellular vesicles are small. They have a bilayered lipid structure. They're released from a parent cell. They're released from all cell types. They're particularly released in large amounts from tumor cells, and those EVs tend to have more mannose on them. EVs are involved in cell-to-cell communication. They carry a cargo, a payload, if you will, with a variety of structures, including microRNAs, that gives a signal to the receiving cell. So why that's important in malignancy is the cargo in the EVs from the tumor cells contributes to metastasis or spread of the tumor, growth of new blood vessels feeding the tumor, as well as microRNAs that suppress the body's immune system. And so the theory is in cancers, if you can remove or debulk the body of those EVs from the tumor cells, you could improve responses to cancer therapy.
Got it. Okay, so that's the right way for us to think about it. These EVs can be are harmful, and you believe the device can help remove them. So let's talk specifically about the clinical trial that on which you're about to embark. Um, if you want to give us, some information about the trial, about how it's designed, that would be very helpful, I think, to the listeners.
Sure. So when thinking about going into, and again, this is our first foray into oncology, thinking about how to structure an intervention, this is an extracorporeal circuit. So it involves putting a vascular catheter in and putting somebody's circulation over a device. So you want to go initially into a population that's not going to just be treated adequately with an oral drug or an IV drug. There has to be of some potential additional benefit to the patient. So with that in mind, what we chose was to go into a population that was receiving immune checkpoint inhibitor agents. So the drugs are that you probably most familiar with are Keytruda and Opdivo. These are immune checkpoint inhibitors that block anti-PD-1, which is a suppressing molecule in cancer. So these drugs have been revolutionary. There are many cancer types for which people their disease would have progressed and they've died, but are now living. But the difficulty becomes only about 30 to 40% of patients who receive these drugs have a lasting clinical response, which means there's an unfortunate 60 to 70% that are going to go on to salvage therapies of unproven efficacy and have a lot of toxicity. So when we thought about structuring a trial, we said, let's pick a population for which they'll get their standard of care with their anti-PD-1 therapy, either Keytruda or Opdivo. They'll get it as they normally would by their doctor for the first 2 months. And we'll measure extracellular vesicles. We'll measure their body's own T cell ability to fight tumors while they're getting that. And then they'll get checked at 2 months as standard of care. And if they're not improving, then we would put them on the Hemo Purifier and then see what happens to their extracellular vesicle levels and their body's T cell tumor fighting ability. And so that's the way it's structured. And so it's structured as a safety and feasibility study. The first important thing always is, does this patient population tolerate this therapy? And so that's the main focus. But built into the study is also, again, what is the device doing to the extracellular vesicle concentrations, the T cell fighting ability? And also, how often do you have to administer such a therapy to have lasting responses on those two factors? So in the Hemopurifier phase of the study, it's done in 3 sequential cohorts. The first cohort gets a single Hemopurifier treatment during a given week. The second cohort gets 2 Hemopurifier treatments in a week, so say Monday and Friday. And in the third cohort, they would get it 3 times a week, say Monday, Wednesday, Friday. And there'll be a stop after each of those cohorts where the Data Safety Monitoring Board will look and make sure there's no dose-limiting toxicity. But we will also get readouts from our central lab on those extracellular vesicles and those T cells so that we could start to make some decisions. We'll follow those labs out for 8 weeks after the Hemopurifier treatment. We'll also follow the patients out much longer, to a year, to see how they're clinically doing. But the trial is a small safety study, so it won't give us the ultimate determination of clinical efficacy, but it will give us the biologic signal and the safety to justify if we want to go on to a further true efficacy trial.
Got it. So the hypothesis is that the Hemopurifier, in conjunction with Keytruda and other checkpoint inhibitors, can move the needle on that 60 to 70% of the population that does not respond long-term to the treatment?
Yeah, so to expand on that, that you're removing extracellular vesicles that are likely playing a role in resistance, if you will. I'll use the term resistance to those therapies, and that by removing these, you could resuscitate the response to those therapies. So yeah, move the needle, take it from a 30-40% to something much better.
Got it. Okay, great. Um, okay, so what stage are you? You've had a lot of news flow recently. What stage are you at right now in terms of the clinical trial, including today's news, which
Yeah.
I don't know if everyone had a chance to see.
Yeah, so we've been very busy, which in the clinical trial arena in biotech is a good thing, right? So yeah, we're having a similar trial being done in two countries. One is Australia and one is in India. So in Australia, we have identified 3 sites that are highly skilled at such trials and who were interested. And we already have one site actually that has gone through the entire process that's open and active for enrollment, meaning they could screen patients today, tomorrow, and when they find an eligible patient, they can enroll. That's at Royal Adelaide, the clinical trials unit there, which has a long history of doing excellent work in this space. So they're open. So the study is open for enrollment in that country with that site. On the heels of that, a second site, Pindara Hospital in Queensland, has also gone through the whole ethics approval and governance process and successfully. And in the next few weeks, there'll be what we call a site initiation visit, which is the formal training on the protocol procedures. And we expect that site will also be open for enrollment before the end of this month. And then we have a third site in Sydney that's going to be that goes through the same ethics committee as Pindara that I just mentioned, that is submitting to be added to the ethics committee approval for Pindara, and that's going on right now. And then that will be followed by governance. So right now, 1 open site, soon to be 2 open sites by the end of the month. So enrollment and screening can occur right now in Australia. On the other trial is in India. It's what Medanta Medicity Hospital, which I think we'll probably talk about more, but has a storied history in clinical trials and in this space. And we just announced today that their ethics committee approved the study. And so we will follow that up very shortly with completing our clinical trial agreement and budgeting, and also the import-export licenses that are needed to ship samples and devices. So we anticipate that they would come online before the end of the year as well. So a lot of activity in the last couple of months. It's very exciting.
Right. Yes, a lot of activity. So you're talking specifically about, doing most of this activity in Australia and in India. So why did you decide to conduct the initial clinical trials in those 2 markets? Can you give us some
Sure,
color on that?
sure. Australia has had a long history of doing important early stage clinical trial work in both in drugs and medical devices, world-class medical hospitals, physicians. They also have a very interesting and helpful life science tax credit arrangement where for life science work, because they want to build their industry in that sector in Australia. So we are participating in that program. We can get up to 43 cents on the dollar back of dollars that we spend in Australia on this clinical trial. And that's not a tax credit, that's actually cash that we would get back one time a year. So that's very helpful in keeping the costs down. And these hospitals that Steve just mentioned are excellent. They've done a lot of clinical trials, and that's why we started there. India, that particular hospital, Medanta Medicity, likewise is a world-class institution. The physicians there are world-class as well. And that group of nephrologists at Medanta Medicity has done probably 120 of the human treatments with the Hemo Purifier. So they're the world's experts in using our device. They're big supporters, and we're supporters of theirs. So it just made sense to work with that group once again. So that's why we're focused on these two countries internationally first, and then we'll turn to the U.S. after that.
Okay, got it, understood. Um, and it sounds like everything is really moving along very quickly towards clinical, you know, um, research very shortly. Are there any other companies that you're aware of in the overall medical device space that currently have other devices or cartridges that are also under development? And if you are aware, can you tell us a little bit about how you see those competitively versus
Sure,
what you're trying to do?
sure. There are several smaller publicly held med device companies that have extracorporeal cartridges like ours. None of them are focused on cancer, but the companies that are often mentioned by our investors and potential investors that they track are Cytosorbents Corporation. Their ticker's CTSO. Their market cap is about 66 million. C-Star Medical, it's about 21 million in market cap. And Spectral Medical, which is a Canadian company, they're about 142 million in market cap. Uh, they're largely focused on inflammation reduction and sepsis. We're not aware of any work they're doing in oncology. There are several other small privately held companies, but in terms of the public potential comps, even though they're not really comps because their mechanism of action is totally different than ours, I would say those three companies: Cytosorbents, C-Star, and Spectral Medical.
So it's fair to say then, based on what you just said, it's fair to say that there really is nothing out there that you would say would directly compete with the Hemo Purifier.
There are no publicly held companies that do it. We're aware of one privately held company that's, again, the product's different than ours. It has a different mechanism of action, but we know of one private company that may be venturing into the oncology space, but there's not much of a competitive field that we're aware of.
Got it. Okay, so now that you're ramping up your R&D activities with the pending launch of these clinical trials, can you talk a little bit about what your position is in terms of your supply of the device, your supply of hemopurifiers, your access to hemopurifiers, production of them, and is if that's something we should be looking at?
We believe we have sufficient inventory for these trials. And I should mention the trials are designed to be between 9 and 18 patients in both countries, 9 to 18 in Australia, 9 to 18 in India. Partially because they have different standards of care in those countries, different regimens, periods of time between Opdivo and Keytruda doses. So, and the demographics and backgrounds of the people might be different as well. So there were two reasons to have two sets of patients rather than combining them both into one overall trial. So we do believe we have sufficient cartridges. Our manufacturing suite that we brought up to speed over the last 2 years is up and running. The FDA approved it recently, and we just did a manufacturing run in the last week or two, and we're planning another one soon. So there's the relevant raw materials are available, and we think we're okay, Marla, from an inventory perspective.
Okay, great. And can you talk a little bit, give us a little bit of color on your balance sheet? I think everyone
Sure.
would be interested in hearing.
Sure. Well, we just reported our June quarter recently. We closed June with about 8.2 million in cash. We have no debt of any sort on our balance sheet. No convertible debt. No debt of any sort. We have approximately 14 million shares outstanding, and a very clean balance sheet. That we're trying to keep it that way. So many small publicly held companies have these convoluted structures with preferreds and convertible. notes, and we have no preferred stock, no convertibles.
Okay. So now that we know you're moving into this next phase in, looking at the Hemo Purifier, can you talk a little bit about what you see as upcoming milestones or potential milestones that we should be looking out for?
Sure. Well, as we recruit patients and start to treat them, we think that's important information, and we'll make that known via press releases or 8Ks. Down the road, we'll start getting data sets from each of these tranches of patients that Steve mentioned. Once that analysis is done by the lab that's doing all of this work for us, we'll make that public. I think a lot of people, including us, are quite interested in that data. So we see a whole series of milestones over the next year plus, Marla. And we didn't talk about the size of the market. I mean, we know everybody knows many people, unfortunately, with cancer. I know my sister died of it a few years ago. It's a large market. We've done a little bit of research on it. If we may, Marla, can we just touch on that?
Please do.
Steve, do you want to
Yeah.
cover
So as I mentioned, immunotherapy agents and these checkpoint inhibitors are really been game changers. And one recent piece of information is that of all cancers, about 40% of people, 40 are eligible for an immune checkpoint inhibitor. So let's just take one of the two agents that I mentioned that are going to be in the clinical trial, Keytruda from Merck. So they're based on their own company's data. They estimate that in 2024, this year, that 2 million patients will be on Keytruda. So if you use the information that I gave you, that about only 30 to 40% will have a lasting response, that means about 1.2 to 1.4 million patients are not having a lasting response. So that's just with that agent alone. So that's a very large population. And then
Yes.
nivolumab has a smaller market share, Opdivo, but again, still a very large population with a similar response rate.
And the number
Well,
of tumor types that are being approved, I think it's close to 30 now
Yeah.
for Keytruda. So those are very successful drugs, even though they only in the end help 30 to 40% of cancer patients. So if we can improve that 30 to 40%, we'll have achieved our dream.
Yes. So can you, based on what you just said, Jim, about, several different tumor types, your trial is designed to look at a range of different tumor types. Is that correct?
Yeah, I should have mentioned it. So it's called a basket trial. So we're agnostic. We're not looking at one specific tumor type. We're looking for at least, I believe we have 8 or 9 different tumor types with very common tumors, including non-small cell lung cancer, which is one of the most common cancers that people unfortunately suffer from. Melanoma, again, a big problem. Mesothelioma is included. Gastrointestinal tumors, both in the upper GI tract as well as the colon, are included, as well as urothelial cancers, bladder and renal cancer. So it is a basket trial. So this trial, and each patient is their own control. So we'll be able to glean in a rather small trial what our biologic signal is in a variety of tumors. Our hope is that exosome removal results in a similar response in regardless of the tumor type, but we'll see. But we are casting a wide net.
So just to make sure I understood one thing you just said, that each patient will basically be his or her control group. You will be monitoring the patient's exosome and EV level versus pre-treatment with the hemopurifier?
Yeah. So
Is
the
that
way it was designed is this run-in period. So these people are getting these drugs as their standard of care, but they're consenting when they come in to have blood draws during their 2 months of anti-PD-1 therapy alone, specifically so we can see what their extracellular vesicle, their exosome levels are, and their T cells' ability to fight tumors is before they fail, if you will. So we'll have a profile of what the EVs and the T cells look like before failure of the drugs, and then does the Hemo Purifier change that? So they're their own control. They're before and after Hemo Purifier.
Right. Okay. Thanks very much. This was very interesting. I think it was, very informative. And if anyone's interested in following up, please reach out directly to the company or to OTC Markets or to us at Zacks. And thanks everyone for participating. This basically ends our presentation.